Hi students, today we are going to discuss about the structure of bacterial cell. If you like my channel, press the like button and share with your friends. So let's enter into the topic, the structure of bacterial cell. So this is the structure of bacterial cell. I hope so it is visible to you. So let's discuss the parts of the bacterial cell. The number one is a cell wall. Number one is a cell wall. Okay. Number two is a cell membrane. Number two is a cell membrane. Number three calyx. Number three is in glycocalyx. So glycocalyx as well as the cell wall along with the cell membrane form cell envelope. Glycocalyx along with the cell wall and cell membrane forms cell envelope. Okay. So where do we find the glycocalyx? So this glycocalyx is a gelatinous layer fixed to the surface of the cell wall. It is a gelatinous layer fixed to the surface of the cell wall. So on the surface of the cell wall we can identify a special structure, a layer called that is a glycocalyx. So this layer is formed of the glycocalyx, a gelatinous layer. Okay. So sometimes this gelatinous layer is not fixed to the surface of the cell wall of the bacteria. So it is loosely packed. Okay, when it is tightly packed or bound to the surface of the cell wall of the bacteria, then we call it a capsule. We call it a capsule. If it is not bound, fixed to the surface of the cell, but it is making a layer, so loosely packed layer, then we call it a slime layer. We call it a slime layer. Okay. The outermost layer, so in case of bacteria, we will identify the outermost layer which is known as an glycocalyx layer, that forms a capsule. Okay. So followed by that outermost layer, followed by the capsule, then we are able to identify the cell wall. The cell wall of the bacteria is composed of the polypeptidoglycan. 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 The cell wall of the bacteria is composed of polypeptidoglycan. So if that cell wall is composed of the polypeptidoglycans with many layers, okay, then they are known as gram-positive bacteria, because this gram-positive bacteria, they respond to the gram staining procedures since it is a thick layer. The gram-positive bacteria cell wall is very thick and they respond to the staining process, give the positive response. But in case if the cell wall is made up of only fewer polypeptidoglycan layers, then it forms a thin layer, and that gives a definition to the gram-negative bacteria. Gram-negative bacteria, because this gram-negative bacteria cell wall is made up of only few layers of the polypeptidoglycan. Okay, so that's why it is very thin. So based on the cell wall and their response to the responsive manner to the gram staining procedure, the bacteria is classified as gram positive bacteria, gram negative bacteria. Okay, so the cell wall is playing a major role to the bacteria because it is giving a definite shape to the bacterial cell. Shape to the bacterial cell. It is very rigid in structure. It is very rigid in structure. Okay. So, and this cell wall of the bacteria playing a major role in all the cases. Biological activity. For the biological activity of bacteria, the cell wall secretes the substances. The chemical substances secreted at the cell wall play a major role in the binary fission or sexual reproduction of the bacteria. Okay, many metabolic activities are controlled by that enzymes or the substances produced from the cell wall. So we are taking antibiotics. Okay, what is the use of antibiotics? Antibiotics works with the bacterial diseases, but it will not work with the viral diseases. Why it is so? The antibiotics work with the bacterial diseases, but it is not working with the viral diseases. Why it's so? The virus is not having that. If you remove the cell wall, if you remove the layer of the capsule of the virus also, and it will be able to synthesize its material and replicate, because the center of the virus is playing a major role. But in case of bacteria, once the cell wall is damaged, the bacteria will not do the proper metabolic activity, so thereby it will inhibit the growth of the bacteria. That is the reason we consume antibiotics continuously 3 days in order to suppress, in order to slow down the work of the bacteria. Once the cell wall is lysed, the bacterial cell wall is lysed by the antibiotics, then no more capability of the bacterial cell to synthesize, to replicate its own DNA, genetic materials, to multiply. Okay, all the things are prevented once the cell wall of the bacteria is lysed. So it is lysed with the help of the antibiotics. But these antibiotics will not work with the viral diseases, because the virus, that's a core of the virus, is synthesizing many enzymes in order to multiply, because this virus is replicate our DNA. That means it mixes with our DNA, genetic materials, making a combination DNA RNA complex, finally viral DNA complex. So this viral, our body system, our blood cells are WBC will not recognize this type of changes, because it thinks that it mimics, the virus mimics the genetic materials, genetic makeup of the organism, so thereby it easily escapes from the WBC cells. Okay, fine. So I am discussing about the cell wall of the bacteria. Once the cell wall of the bacteria is lysed, once the cell wall of the bacteria is lysed or damaged, then it will not able to do any activity, because the cell wall is responsible for synthesis of essential enzymes for the metabolic activities of the bacterial cell. Got it? So next things, followed by the cell wall, then we have another membrane that is that's called the plasma membrane or cell membrane. The extension of the cell membrane forms a specialized structure called mesosome. It may be the tubular structures, it may be the spherical structures, whatever it is. So the extension of the plasma membrane forms a special structure called mesosome. What is the function of the mesosome? What actually it does it? The mesosome play vital role in the bacterial cell. How? Because the mesosomes help in the replication of the DNA process and distribution to the daughter cells. As well as they are involved in the cell wall formation. Cell wall formation. So these mesosomes are involved in the cell wall formation. Along with that, the respiration process, secretion process are done with the help of the mesosome. So this is the fundamental and the vital role of the mesosomes. The mesosome is involved in the DNA replication and cell wall formation, as well as the respiration, secretions. Okay, clear? So this is mesosome. Okay, fine. Next things, we are able to see certain structure called ribosome. Certain structure called ribosomes. Okay, in bacteria, the ribosomes are present in 70S manner. So 70S manner. So what is 70S? Yes, what is the S? Swedberg unit of the DNA. Okay, so 70S. So out of this, because this ribosome is having two subunits. One is a larger subunit, another one is a smaller subunit. The 70S of smaller subunit is 30S. The 70S of larger subunit is 50S. The 70S of the smaller subunit of ribosome is 30S, and the larger subunit of 70S is 50S. Got it? Okay. So certain cases, this ribosomes, 5 or 6 ribosomes attached with the mRNA forms a polyribosome or polysome. So this is, we call it a polysome. Polysome. So here you will be able to identify. So 4 or 5, more than 5. Okay. So 5 or 6 ribosomes are attached with the mRNA forms polysome. So you know what is the function of the ribosome. The ribosome is the site of protein synthesis. Got it? Clear. Okay. Certain cases we are able to see some tubular structures or vesicle formations. So these vesicle formations we know as a glycogen vacuole. Okay, glycogen vacuoles with glycogen granules are present in the bacterial cell. Okay, fine. So next things we'll move on to the DNA. Look at the DNA. In case of bacterial cell, the DNA is a single circular DNA duplex. Single circular DNA duplex. Okay. So it is formed. It is in case of bacteria. This is, we call it a nucleoid. Okay. There is no nuclear membrane. There is no nucleolus, and all these things are infused. So forms a circular DNA duplex nucleoid. Okay. So the genetic materials in the DNA, in the bacteria, we call it a nucleoid, since it is not able to distinguish properly. Okay, fine, that's good. Clear. So on the surface of the plasma membrane, we are able to identify another granules or that substances, we call it in respiratory enzymes. Respiratory enzymes. Okay, on the base of the plasma membrane, we are able to identify 3 different structures. One is an flagellum, another one is an pilus. Okay, flagellum, pilus. Clear? Another one is an fimbriae. So on the surface, on the base of this plasma membrane, we are able to, so extension, so from the extension of the plasma membrane, then the 3 structures are arises. Okay, they are tubular structures, thin and long. Clear? Some are short. Some tubular structures are short. So first one is an flagellum. First one is an flagellum. Okay. Second thing you are able to see pilus. Okay, the third one is an fimbriae. What is the differences in the flagellum, fimbriae as well as the pilus? Okay, look at these structures of the pilus and flagellum, fimbriae. Fimbriae is a long and thin tubular structure. Okay, in case of pilus, it is very short tubular structure. In case of flagellum, it is having composed of 3 parts. One is a basal body, another one is a hook, third one is a filament. Third one is a filament. Got it? Third one is a filament. So together it form the flagella. Okay. So what is the function of the flagella? It is helping to swim. For the motility, certain bacteria are motile, certain bacteria are non-motile. The bacteria which are motile, they use for the locomotion purpose, the flagellum. Flagellum are used for the locomotion purpose. Clear? So next thing is in pilus. Pila. So what is the function of this pila? It is a very short tubular structure. The pila is helping for the conjugation process. For the conjugation process, bacteria undergoes 2 modes of the reproduction. One is in asexual mode and sexual mode. In case of asexual mode, the bacteria undergoes the binary fission. Okay, so in binary fission, the bacteria is divided into 2 equal parts, and divided parts develop into daughter cells. Okay, that's in asexual reproductions in bacteria. But sexual reproduction in bacteria also take place through the conjugation method. Conjugation means connect. One bacteria have the contact with another bacteria through the bridge or tubular structures called pilus. So through this pilus, the genetic materials are exchanged between the bacteria. Clear? That's the function of the pilus. So the pilus, the structure acts as so it is known as a sex pili. Okay, because involved in the sexual reproduction of the bacteria. So sex phylae. The third one is a fimbriae. This fimbriae, it helps the bacteria to attach with the solid surfaces. Helps the bacteria to attach with the solid surfaces, so that bacteria is able to attach to the solid surfaces, remain rest at sometimes. Got it? Clear? Do you understand what are the things? So first thing we have to remember mesosome function, and then pilus function, then what we have polysome. Clear? The cell wall function. So cell wall, polysome, pilus, then mesosomes. Glycocalyx, already you know that if it is packed, bound to the surface of the cell, the gelatin layers, then that forms an capsule. If it is not, it is loosely packed on the surface of the bacterial cell, then we call it a slime layer. Clear? So thereby we are able to understand the function of the bacterial cell wall. The bacterial cell wall has the site of production of various substances which is needed for the cell activity. Once it is damaged or lies, no more the bacteria is able to do any metabolic activity. The growth of the bacteria will slow down. Finally, the death of the bacteria will take place. Got it? So thereby we are able to cell wall, site of important synthesis. Okay, then mesosome, replication of DNA, distribution to daughter cells. Respiration, secretion, mesosome. Okay, then by conjugation process. Analogous to the pile is analogous to the sex organ. Okay, conjugation process. Then what do you call it? Fimbriae. Attachment. Attached to surface. These are the things you have to remember in case of bacterial cell. Okay. What is the inclusion bodies in bacterial cell? Inclusion bodies, so the glycogen granules, lipid granules, polysaccharide granules, all these things are present as in granules as well as the vesicle form or tubular forms inside the cytoplasm of the bacteria. That is, we call it inclusion bodies. Okay, so we know already about the ribosomes of the bacteria. It is having a 70S. Okay. So ribosome is known as the site of the protein synthesis. So we are in case of the bacterial cell, the ribosome is in 70S form. Okay, so having the two subunit, smaller subunits, larger subunit. The smaller subunit is 30S, larger subunit is 50S. In case of eukaryotic cells, the ribosomes will be having a 80S. Okay, so we will be having, so that is a 40S as well as 60S. But these are the things regarding the bacterial cell. So you have one questions. Do viruses is composed of cells? Do virus is composed of cells? No, the virus is not composed of cells. So what is the nature of the cell? The cell is a fundamental unit of life, structural fundamental unit of life. It does its work, okay, independently. The cell is independently existence. But in case of virus, when the virus is in the atmosphere, it is considered as a non-living thing. When it enters into the host organism, it became activated. So it became activated and utilized the host organism genetic materials to escape from this host organism defense mechanism. Okay, so thereby the virus is not composed of the cells. Okay, because the virus is dependent. The virus is not an independent existence, can't do self-replication, can't do self-reproduction, can't do self-activity. They are requiring the help from the host organism body. So thereby it is doing its work. Okay, so thereby we can say that virus is not composed of cells because the virus is dependent. Okay, and doesn't have the ability to do self reproduction, self mechanisms. Okay, that's good. Thank you.